It is a pleasure to welcome you to the third edition of the lecture series on advancements in geotechnical engineering, from research to practice. The AGERP lecture series is a pro bono initiative led by Dr. Partha Mishra and Professor Sarat Das. Initiated in 2020, it is aimed at disseminating the coupled learnings from academia and industry on some of the key topics in geotechnical engineering. The International Workshop on Biogeotechnics was hosted in 2022 during the third edition of the AGERP lecture series. The following lecture on stabilisation of iron waste, a role for biogeotechnics, was delivered by Professor Gordon Southam at this workshop. Professor Southam received his BSc, Honours, and PhD in microbiology from the University of Guelph. Following his postdoctoral education funded by Western Mining Corporation, Australia, Southam accepted a faculty position in the Department of Biology and Center for Environmental Sciences at Northern Arizona University. He joined the Departments of Earth Science and Biology at the University of Western Ontario when he was appointed Canada Research Chair in Geomicrobiology from 2001 to 2011 and Director of the Center for Environment and Sustainability from 2010 to 2012. In 2012, Professor Southam joined the School of Earth Sciences at the University of Queensland as the Val UQ Geomicrobiology Chair, and is currently Professor of Geomicrobiology in the School of Earth and Environmental Sciences at UQ. Professor Southam is an interdisciplinary researcher who has crossed traditional boundaries between biological and geological sciences. His research takes fundamental geomicrobiology and translates this into applied settings, primarily in the minerals industry. Professor Southam's landmark contributions have been in understanding the fundamental roles that bacteria play in catalyzing the formation of economically significant geological and mineral phenomena, such as the formation of placer gold, their role in mineral carbonation, CO2 sequestration, bioleaching of metal sulfides and bioremediation. Reflecting the interdisciplinary nature of his research, Professor Southam has published 204 articles in 89 different refereed journals.
So I'll start off by kind of featuring Alan, Anat, and Emma, who did the lion's share of the work that I'll present today, and it's inspired by Alejandro that I need to be speaking to engineers a little more than I have been, and so I'll be looking at an iron mine waste site, and hopefully I'll demonstrate a need for biogeotechnics. So as an introduction to me, this is the kind of thing I look at. We're down at the micron scale. We're looking at bacteria, kind of the environments that they see, that they experience, and certainly this one here has been completely mineralized. And so if you live by diffusion, it's hard to get nutrients in and out, and so we've got a fossil on our hands here. Now, when I present to microbiologists, I default to being a geologist, though I've never taken geology before. And when I hang out with my geology colleagues, well, then I become a microbiologist. With an engineering audience, I'm not going to stray into your field, but I'll stick to microbiology and geology and see where we go. I did, after agreeing to give a talk on biogeotechnics, I thought I should maybe, when I was then asked to give a topic, go to Google and see a little bit more about what you're about, and Alejandro gave a great background there. And so this idea of studying and using processes, and the word that kept coming up was stabilization, slope, soil, subbase, subgrade, surface erosion protection. The stabilization was key, and we've been interested in stabilizing iron mine waste from the standpoint of mine site remediation. But this idea of stabilization certainly is important throughout geology, in particular with respect to tailings disposal. And so that's certainly an area that we're interested in. Now, I'm going to take you to the field, because one of the advantages of being an environmental microbiologist or a geologist is we go to some of the most amazing places in the world. And so this is my phone shot out the window as we are coming into land near Carajás and Para in Brazil. And so we're in the Amazon rainforest, and the rainforest in this environment represents the topographical lobes. From the mine site perspective, we are interested in the plateaus up at the top of this image here. And so when you land in Carajás and you drive to the iron mines, this is the kind of environment that it changes to. And so we are literally in the middle of the Amazon rainforest, and you're looking at a dry, arid, scrubby desert kind of situation. In the top right there, you've got the plateau that this photo was taken on. We're looking at square kilometers in area. This was actually the Discovery Plateau that somebody in a plane, kind of 80 years ago, was flying over this flat, hard environment and noticed it was different and landed the plane and broke off a bit of the iron oxide and tossed it in the plane and took it out. And so these are the iron mines. And so the iron mines there are on the scale of kilometers. When you go to the edge of this plateau, that's the photo on the top left at the top. And so there's erosion along the edge, and there's massive blocks of iron oxide that are falling down into the rainforest that continues hundreds of meters. And so we're interested in this environment here. From a mine site restoration, can we reform Kanga after mining is complete? And is there a role for biology? And so when they mine, this is the sort of thing that you see. That upper surface, there's some grasses and there's a few scrubby trees. It's just described as an iron duracrust. It's hard, it's cemented. In Brazil, it's referred to as Kanga. And the cements up there are primarily goethite. So there's a range of iron oxides in here, and it's supposed to be a dynamic self-healing process. Below this duracrust, the silica has been leached. So the original formation of an iron deposit has alternating layers of iron and silica. They're associated with early Earth events as we oxygenated the planet and literally rusted out the oceans. And so seasonally or probably likely due to seasons, every time you have growth of biology, you have production of oxygen, and the oceans rusted out and it's settled. Now, in the iron deposits in Brazil, these surfaces are up to 5 million years old. And while you can only put about 20 ppm silica into solution, all of the silica is leached out, almost producing pure, soft and question marks, iron oxide. And so the latest mine in Carajás, S11D, they've actually stopped using trucks. They're just going in with front end loaders. They're digging up the iron oxide. They're throwing it on conveyor belts, and then they're loading rails to take it to market. And we're interested in this duracrust at top. Once we've done mining, can we reestablish this unique ecosystem? And so this question of geology versus biology was presented by Dorr in 1964. We're looking at almost 60 years ago. Dorr said that this is too weird to be geology and that there must be biology involved in some capacity. And so here were the features of Kanga that were described in his paper, that it's porous, but it's resistant. It forms at the ground surface, so this idea of needing oxygen. It has lots of fragments of hematite, but it's cemented. It can include soil, but the soil is really being generous. It's more just iron oxide. We've got this idea of porous limonite. Limonite is a junkyard term that says it's kind of rusty and it's an iron oxide, but we're primarily looking at goethite. Kanga is described as having a high amount of phosphorus, 0.09% on average. And we asked initially, is 0.09% phosphorus significant? And if you kind of apply it against the Redfield ratio, which is kind of an algal molar quantity, 1 gram of phosphorus is enough to produce 3.2 grams of biomass. All right, so this idea that 0.09% phosphorus is plenty of phosphorus to grow biology. And so we sought about seeing if we can demonstrate initially, can biology be involved in the Kanga formation? And so here we are back on the plateau. There's patches there where there's really nothing growing at all. And strange looking bromeliad type plants and the grasses that are calling this home. Remember that this dry arid environment receives meters of rain every year, right? It's in the rainforest, it gets the same amount of rainfall, but it is not supporting rainforest biota. If you zoom in on Kanga, this is the kind of thing that you see. You can see the kind of the limonite texture, the rusts that are there. I describe this as organic looking rock, which the geologists really don't like. But you know, I'll ask Alejandro later if he'll back me on this. We've got all of these layered textures that are actually similar to stromatolites that grow layer by layer. In the case here, though, it's iron oxide. If you break off a piece of this rock and polish it, so here's a 30 micron thick section of a polished rock. You can see the layers, you can see the grains that have been cemented together to one another, and this idea of a layered stromatolitic type growth, although not involving carbonate, in this case exclusively involving iron oxide. So if we zoom into this polished rock with backscatter scanning electron microscopy, we've got kind of aluminum and iron-rich horizons. You can see all of the clasts in the material kind of in the background that are cemented by this. If we zoom into to the iron oxide here, this is what we see. We've got iron and alumina-rich fossils throughout all of the cements. There's certainly some evidence of chemical oxidation of iron, which could happen in these kinds of environments, but fossils are integrated in the formation of these cements. If you zoom in, you can appreciate the diversity. That bacteria typically have a consistent, they can vary a little bit in length, but they'll have consistent diameters. And so just looking at diameter alone, we've got a diverse ecosystem that's responsible here. One of the things that we were able to do is, it's in kind of the long rectangle just below the text there, is with an ion beam, we can dig into the surface that contains these fossils and lift out a section of the bacteria to get a new appreciation of biodiversity. And this is what we saw here. The entire field is the ion milled bit that was pulled out. You can see the different cell envelope structures, not just different diameters, different chemistries involved here. In the bottom left, there's some evidence of chemical precipitates that are held together by the biology. And we went looking at kind of the 8 micrometer box from the standpoint of, are we just looking at structure here, or is there chemistry that tells us that we've got biology going on within these samples? And so here's the nanosims that we've done, and we've got carbon associated with the walls. We've got carbon-nitrogen bonds that are consistent with protein. We've got phosphorus in the cell membranes that are consistent with phospholipids. The fossils themselves are actually iron lows. There's certainly a large iron matrix, but what was remarkable is that we had alumina highs in the fossils, and the idea of aluminum being mobile under circumneutral conditions is kind of a strange feature of Kanga. Now, the Kanga itself isn't just these kind of organic looking rocks. There's the grasses and trees, and from the convenience of being able to pull these things out, we were focusing on the grasses. And so if you notice here, there's a dimpled texture over this, the edge of this field here. If we take a zoom in, you can see that the grasses are attached to the Kanga. And in fact, all of these dimple textures are relics of previous plants that have dissolved their way into the iron oxide surface to get a hold of the surface to persist, but also likely to get phosphorus out of Kanga to support the growth of the plant itself. If you go into one of these other areas, so we were interested in kind of the areas where there was no plants. We had a small rectangle at the top there above the text. We broke off these kinds of materials and we polished it the same way that we had polished Kanga. In this case, though, looking at a cross section through the Kanga where plants had once been. And this is the sort of thing that we saw, and we found it quite surprising. In panel A there, we've got 2 of these dimples where the plants have literally grown into the surface. The main matrix of it is kind of an iron alumina silicate versus the pure iron that's producing these cements. If we zoom in looking at backscatter imaging, where we find the iron highs in panel D, we're finding fossils. So we've got a strange phenomenon here where the plant microbe ecosystem is both dissolving Kanga and reprecipitating Kanga. And if you're looking at this self-healing property of that's attributed to the Kanga environment, that this idea to dissolve and reprecipitate iron becomes critically important in these kinds of environments. When we looked at the plants that we had kind of pulled out of the dimples, there's certainly they're staining with iron, and there's evidence of iron oxides amongst the plant. And if we zoom into those, we're getting the same kinds of diverse microfossils. In this case, that's completely cemented, where the outside as well as the inside of the bacteria have been preserved in these environments. And the dark regions in backscatter corresponds to the organics. The organics that template the iron oxides to grow on the outside or the inside of these cells. Now, one of the more unusual things that we saw at S11D was that the surface of Kanga appeared to be soft. And if you break off a chip of this, we can see at the bottom there that we've still got our layered iron oxides, but we've got kind of a biofilm growing on surface. And we were interested in this biofilm to see whether it was, dissolving and reprecipitating iron the same way that we'd seen the kind of the earlier kind of plant fossil systems. When we zoom into this texture, we've got cyanobacteria that are growing in this environment and precipitating iron. And so the formation of oxygen in these environments would certainly favor that as a process. And the other organisms that we found dominating it were fungi. And fungi are very good at degrading organic carbon and recycling things. They produce a whole range of chelators that can help dissolve iron oxide. And so we've got kind of it's a variant on a lichen, where we've got a balance between photosynthetic bacteria and fungi that are achieving the same kind of phenomena that the plants are, where we've got dissolution and reprecipitation of iron forming these cements. And we look at the biofilm on surface here, we've got kind of an angular texture on the left where the biology is breaking down. And we've got evidence within the iron oxides that we've got the same kinds of fossils that are infilling. So we've got two biological processes here that are responsible for this dynamic dissolution, reprecipitation of iron in Kanga. And so the key kind of iron reaction sequence here, one is anaerobic. So we've got iron reducing bacteria. The simplified reaction with iron here, we've got an electron donor. There's a whole range of bacteria that will be involved in anaerobic respiration. The more likely reaction, though, is that involved with goethite. And so we've got a simplified organic acid there, and we're taking our iron oxide solid, and in using the iron as the electron acceptor, we're producing iron 2. And so we've got a link between iron 3 to iron 2 to dissolve the iron oxide. And then the idea of rusty films or coatings or biofilms, well, that's easy, right? It can happen chemically, it can be abiotic, and it can also be accelerated by iron oxidizing bacteria. And so the oxidation of iron back to iron 3, producing iron oxide, is a much easier component to this process. So one of the other things that we saw in Kanga was everywhere there was water, there was a metallic film. And so the only way you're going to get a metallic film here is you've got to be feeding the surface film with iron 2 from deeper within the environment. And certainly there was very little oxygen in the water here that the metallic film completely sealed the lower ecosystem from the upper ecosystem. If you take that film and embed it in plastic, and in this case, we're going to cut one of those ultra thin sections, this is the kind of thing that you see. So it's been embedded in plastic, it's been sliced with a diamond knife, and we've got a 70 nanometer slice through this metallic film. There's evidence for some chemical precipitation of iron. You can see kind of the colloids, the dark grains in the upper right and lower left, but there's a whole range of bacteria, all of which have precipitated iron on their surfaces. And so we've got this a new cement layer that's been formed with the aid of biology. Now, Lake Vialao in near the S11D mine gave us a wonderful environment. Not only engineers are inspired by nature, biologists are also inspired by nature. And this provided an excellent opportunity to picture the processes that are involved in forming Kanga. And so we're right at the edge of one of the plateaus. We've got a patch of forest here, and certainly growth of the forest is going to add organic carbon into the ecosystem. And wherever we found water that was flowing out from kind of those little pools, or they're almost like pipes plumbing this system, it contained iron too. So the addition of organic carbon was supporting anaerobic respiration, taking iron 3 to iron 2, and the iron 2 was flowing out from the forested region. Once it reached the surface of the Kanga, along it's a lake here, the iron was able to oxidize. Oxidize both chemically as well as biologically, and as it does, you can see the rusty coatings that are there. Emma, who's there up in the photo, she isolated here, Tomatospirillum, genus of organisms, and in culture, this thing could produce 30 millimolar iron too. Just incredible amounts of iron were put into solution using a mineral as an electron acceptor, which is not an easy ask for this organism. And as it flows out and is exposed to oxygen, the iron oxidation itself was fairly easy. Now, if we look at these films, we saw the same thing as we saw in the metallic film before. That it's loaded with fossilized bacteria. If you take where we're standing here and looking at this image and turn around, sure, I'll come to that in a bit. So here's the Tomatospirillum that Emma isolated from this environment. It's got a diverse approach to growing on iron. It can reduce sulfate. So technically it could be called a sulfate reducing bacterium, and the hydrogen sulfide produced by this organism can chemically reduce goethite into iron 2. It's got a terminal reductase in the membrane that may be passing electrons directly to goethite. And it's certainly producing acid byproducts that will enhance just in general mineral dissolution in these kinds of environments. So here's the photo I thought was coming, where you turn around. And so we have a natural experiment here where we're making this idea of a layered structure of Kanga, just flowing out of the forest and going down to a local lake. Now, if you look at the rock hammer in the middle there, if you break off old bits of Kanga in these kinds of environments, this is what we found. Okay, it's dominated by bacteria. All of the holes in porosity in this environment contain fossils. We got fossils in thin section here. In the circle, you can get more of a 3D view that's provided by kind of some secondary electrons, where the bacteria are infilling these kinds of environments. So wherever water flows, you've got the capacity to support life, and wherever there's porosity here that contains water, we're seeing bacteria. If we zoom into one of these holes, you can see the bacteria. They're growing, they're dividing, they're infilling this structure, and over time you could envision that that porosity will change. That as the holes fill up, the water will be forced elsewhere, and so iron oxidation in the fossils will grow in these different environments. One of the things that should kind of excite you engineer types is shown here. That we've got a matrix of the kind of the iron alumina silicate material that's weathered, and what we've got here is 2 generations of biofilms that are effectively pinching off this pipe that's allowing water flow, and you could imagine that in another 2 or 3 generations, possibly annual seasons, with the massive rainfall that goes into the rainforest, you would expect there to be boosts of activity. The hydrology of this system is going to be dramatically changed by these mineralized biofilms. Now, the challenge that we were presented with is, can the Kanga or duracrust at the top of kind of this edge of the mine, which is typically stored as waste because of the phosphorus. 0.09% phosphorus is not only plenty of phosphorus to grow microorganisms or to support the growth of plants, it's also enough phosphorus to make steel brittle. So picture the Titanic. If there's one thing we don't want steel to be, is brittle. And so while sometimes Kanga can be blended in to the high grade ore down below, in many cases it's now not being used because it reduces the quality of the product. And so you have Kanga stockpiles that are blasted and broken up and torn off the surface of this before mining is allowed to persist. And so the challenge here is, can we use biology to reform Kanga? We've certainly shown that bacteria are involved in Kanga formation. We've shown that bacteria are involved in the iron cycle, both dissolving iron oxide minerals as well as precipitating fresh iron. But the challenge that we have here is that the surface of Kanga, this organic looking material, is some of the oldest surfaces on the planet. That this has been exposed to air using potassium argon dating for up to 5 million years. And so this is an ancient surface. The fossils that we're looking at can be 5 million years old. And so in the Amazon rainforest, you've got 6 months, right? That you can lay out a whole layer of Kanga and try to cement it. But if the monsoonal rains hit you before you produced enough cement, you'll wash away you'll wash away your experiment. And so we're really pushed for time. And when you're pushed for time, you accelerate processes by increasing biology. Bacteria are wonderful catalysts. And so we want to push the system. We've got to inoculate large bunches of organisms. And so the challenge here is, can we reform Kanga after mining is complete, after the surface has been ripped off, blasted, crushed, and stockpiled for decades? Can we bring it back and reform this ecosystem? There are plants here that are only endemic to Carajás, to these plateaus. And so if we can't reform Kanga, then we're going to lose this ecosystem from the world. And certainly the license to mine is going to go away. That part of the responsibilities of the mine companies are to reform ecosystems after mining is complete. And so we're looking to contribute in this area. And so Alan Levitt did an interesting experiment. He looked at kind of a bench scale bioreactor, just on the order of a few decimeters. He looked at Emma's tomatospirillum as the iron reducer. He had a little kind of liter scale bioreactor here. He managed to produce about 80 ppm iron 2. He was looking at millimeter scale Kanga, just that he had looked at kind of a finer fraction to increase kind of surface area and reactivity. And he was pumping in nutrients daily. And so he'd add organic carbon. We had a thick biofilm on the surface of the Kanga, and beneath it, it was anaerobic and reactive and putting iron in solution. And so we just allowed it to overflow by gravity. And eventually it penetrated into the Kanga material, into the reactor. And at the bottom, we would have runoff that would outflow. The runoff never had any iron. So the iron in this reactor was entirely trapped by the processes. And so kind of in that overflow, we could see evidence of kind of the iron film, similar to what we were seeing in nature. And Alan ran this experiment for I think about 6 months. When he finished the experiment, he brought me down to the laboratory and we're having a look at it. And he was really concerned that it looked kind of very delicate, and he didn't want to ruin the samples, and was concerned about how we needed to collect the samples. And I said, well, you just do your best and we'll see what you come up with. 5 minutes later, he called me back down to the laboratory that he was having trouble getting samples out of the artificial slope reactor. And in the end, he needed to use that hammer that's in the photo and a chisel to break off materials. And so here's a piece that was broken off. You're looking at kind of about 3 or 4 centimeter in diameter material. It's grown out of millimeter scale Kenga, and it's completely cemented. I had Alan do a mass balance, and so he cemented kind of cubic decimeters worth of material with only 2 grams of iron. When you look at the flow, the volume, the concentration of iron that's in there, we've only got a couple of grams of iron that have cemented probably about 10 kilos of millimeter scale Kenga. When Alan embedded this and we looked at it with backscatter electron microscopy, this is the kind of thing that we see. So we've got a 100 micron size bar. These are the millimeter scale pieces of Kenga that were part of the bioreactor. But what was new here were these draping textures that were cementing and connecting these grains. We've got kind of spot welds here that are less than 100 microns. If we zoom into one of these, I'm looking at this, and please correct me if I'm wrong, but I'm thinking this, you know, maybe I should tip my computer upside down. Looks more like a suspension bridge. That we've got biofilm here with producing these draping textures that are connecting one grain to the other, and are producing just phenomenal strength in this. If you kind of squint at your computer, you can find that this draping texture is actually porous. It looks more like a sponge. It's composed of iron oxide. If we zoom into this material here, what you see is it's completely fossilized by bacteria. So we've got biofilm that's growing here, that's cementing grains, that's infilling all of the space between the bacteria. The bacteria themselves are still hollow. Some of them may be alive, but they're continuing to oxidize iron to produce this, this structure and strengthening the material. Got one last experiment to share here. One of the things I didn't highlight enough in Alan's experiment is that the bacteria that were within this environment were different than the ones we inoculated. And we were disappointed because we're really kind of betting on this Tomatospirillum as being an important organism. We used it in a subsequent meter scale pilot experiment, and the Tomatospirillum was gone again. That all of the bioreactors here were taken over by a Petitia bacteria. It's a superphylum. It's known to have an active group of iron cyclers. And so we've been surprised by this. We've been doing mineral carbonation work in South Africa, and we're seeing the same kinds of things, that in these diverse ecosystems, the individual organism that comes up in these bioreactors can vary from experiment to experiment. But the chemistry that you're looking for is invariably looked after, that if you create a selective pressure condition that will drive chemistry in one direction or another, that there will be organisms in these environments that will catalyze the reaction. So we'll look first at the pilot scale Kenga experiments. So we threw in kind of a cubic meter, about, I would say each one of these probably had about 1,400 kilos of Kenga that we shoveled in. And the other one that we'll take a look at is the bioreactor. Over on the right, you can just kind of see it on the edge. That one there has half full of media, so about 500 liters of media, and had about 800 kilos of Kenga that was kind of added to the bioreactor using a trowel. And so this was the bioreactor we added the Tomatospirillum to that changed. You change the system, you change the selective pressure, you change the bacteria, but not the chemistry. So we were dissolving massive amounts of iron here and adding that into the bioreactors. And we'll come and look at both of these two systems. So here's the top left is we've cut the face off of one of these meter squared bioreactors. Anywhere near the top, you can break into it and lift off clasts that have been cemented together with both roots, but primarily iron oxides. Throughout the entire bioreactor, we've got Tom there. It wasn't strong enough that we needed a hammer and chisel, that we could break off bits with a pick. But looking at all the materials here, they had fine coatings of iron oxide, as well as some of the biofilms that were very friable and certainly wouldn't have been adding a lot of strength to this system. Geochemically, it didn't matter what we did. If we inoculated or not, if we added carbon or not, if we added plants to some of them. In most of the situations here, the rainforest in the background just naturally seeded these as environments. So from a plant perspective, they were considering this was a soil that basically they all did the same thing. They all cemented Kanga. So we just have to feed these systems, and the natural organisms we're hoping will be able to take this off. And so the last image that I want to leave you with is this one here. That once we dumped the water out of the bioreactor, the Kanga in the bottom looked like cement. And so I'll ask you whether you agree that this looks like cement. I threw the cement into this. It was wet, it was sloppy, and by the time we finished work on the day, it had set. And we have produced from the bioreactor solids a ferrocrete here. We're not at the point where we've got the kind of organic looking Kanga. You can just see the individual clasts of Kanga here, but we haven't processed this material yet, but it's certainly going to take a hammer and chisel to be able to sample. And so I'll just leave that picture on here. We're almost at the point to try a field trial. We've got botanists from Valley who we're working with, and it was literally right before COVID hit. We were supposed to be in Brazil, and we're just not on international travel yet. Soon, and so we hope to get back to the rainforest and try a field trial, not only with the bacteria that we've got there, but also seeing if these wonderful endemic plants can grow in this material.
Okay, yeah, nice. Like if we can go from lab to pilot, that itself is a big deal for, and then from pilot to field, it will be.
Well, yeah, so it's, I spend a lot of my life thinking about things on the micron scale. And so just for me to get to a meter scale, we've increased, 1 million-fold in terms of size. And so I'm interested in whether we can go from, even if we can just increase another 2 orders of magnitude, right? Get to kind of 100-meter scale. I think we'll be on the way. We need to achieve kilometers, so we're 2/3 of the way there. Yeah. So the bioreactor that we in the experiment there went a year. So we kind of had it covered during the monsoonal rainfall. We need to push it. We need to get to 6 months. And so the thing that was surprising was that kind of the bioreactor, just the sludge in the bottom of it was all Kanga, and it looked like cement. I'm wondering here, and stand corrected, is that when you look at concrete, right? We've got the cement that's there. That's a very small fraction relative to the sand and the clastic material that goes in. And so I think we need to take lessons from concrete engineers on how to make concrete and switch it into an iron variety. That the idea that we should be able to cement this material with very little iron is supported by Alan's experiment, right? It took a hammer and chisel to sample something that was less than, kind of 0.2% iron or secondary iron oxide. And so if we can achieve, kind of high strength with minimal cement, then all we have to do is keep it from washing away. Yeah. Right? That if one season can get it to that point, then in subsequent seasons, it's only going to get stronger.
Yes.
And so the goal here is that, if nature might have I don't know, 10 to the 6 bacteria per mil, if our cultures can be 10 to the 7 or 10 to the 8 or 10 to the 9, we can increase reactivity 1,000-fold, or flip it, we can decrease time 1,000-fold to achieve this kind of stability of the waste.
Yeah, true. Even the second question is from my side as well as from his, like, which kind of microorganisms you use? Like, do you go for a single pure culture, or it's a mixed culture isolation?
Mixed cultures, yeah. So we're going for consortia here. There's no, you can certainly isolate pure cultures in the lab, and Emma was a microbiologist by training, so she did that, so she could characterize the organism extremely well. But you can't keep nature kind of sterile. And so this idea of doing field trials, that there's no point trying. The grasses that were in that one photo, we didn't add any of those. It's self-propagating plants that found this surface and thought, yeah, I can grow here, and the seeds germinated and the grasses grew. We've seen that we're looking at weathering kimberlite in South Africa, and in the kind of the non-inoculated systems, we're at over 2 years, and it just looks like rock. We have an IBC that's full of rock. In the biological system, it's covered with grasses again, not planted, not seeded, self-propagating seeds that have just blown in, found the biologically weathered system, and are making a go of it.
Oh, so in that system, do you supply some of the nutrients or even no nutrient?
So some of the controls had no nutrients, and certainly the cements weren't as strong. That what we're seeing with a lot of these organisms here is that fermentation is important. So the idea of organic acids reducing the iron oxide in there. So in that case, we were adding glucose. So just a very simple, just a simple substrate. They get their phosphorus from the kanga, and there's a whole range of organisms there that could fix nitrogen. So all of the basic building blocks that they need for life is present. We just bump the population by adding glucose. We were adding kind of grams per liter quantities. So kind of typical of bacterial culture media that we wanted to push the system. And so what we were hoping was that we'd achieve the same kind of thing. What we're relying on in all of these environments, right, is that we need both anaerobic conditions for fermentation or anaerobic respiration, and we need aerobic conditions. And so part of that is captured with, it wasn't a continuous flow bioreactor. We'd throw in kind of a nutrient or amendment and just let it percolate in. And so you're going to have anaerobic and aerobic microenvironments in this thing. And so we rely on that from a bacterial perspective. What I say to my students is, like, if you're measuring pH in an environment, you've got a probe that's maybe a centimeter, right? If you were to personify yourself as bacteria, that probe is now 10 kilometers, right? So the microenvironments are essential for the growth of all sorts of organisms. And in this iron cycle here, we seem to be getting both dissolution and reprecipitation. Now, both in Alan's project and Emma's field scale, we did employ a bioreactor. And the reason for that was intentionally to separate the processes, that we could enhance dissolution in one, and then by not feeding another one, enhance iron oxidation. And so whether we can achieve it in a one-step process or whether keeping the two processes separate, right? I'm picturing a hillside, right? Like something on the scale of hundreds of meters, where we can have a series of bioreactors that are basically flowing down over this material for slope stabilization. So in that one kind of experiment where we had kind of those draping textures that were coming across, so we've seen that before in coral reefs, in beach rock formation, certainly on Heron Island here, that you can have biology that will grow as a biofilm. It might be on the surface of water. There might be, water held by capillary forces. And so you can literally grow a biofilm on that water-air contact. And when they dry out, they kind of stretch a little bit and you get those draping textures. Now, we've got a system where we're adding in iron 2. So the bioreactor there, we're feeding it iron 2, it's in solution. When that's exposed to oxygen, there's the potential for chemical oxidation, but we didn't see that. That wherever we found the new iron oxides, they were exclusively associated with bacteria. And so bacteria don't get a lot of energy from oxidizing iron, right? You take iron 2 to iron 3, you get 1 electron. And so if you're going to make a go of it as an iron oxidizing bacterium, you've got to oxidize an awful lot of iron. And we think that's responsible for that infilling surrounding all the bacteria. One of the other things that might have played a role is exopolymer. That bacteria that grows biofilms produce EPS. And so the EPS itself might be acting as a nucleating agent. And so you got EPS surrounding the bacteria. And so you then surround the bacteria with the iron oxide. That both bacteria as well as the EPS tend to have a net negative surface charge. And so it will naturally bind cations. And so the iron in the system would naturally stick. Absolutely. So we've looked at asbestos mines, nickel mines, and we're looking at kimberlite in South Africa. So there's a neat environment we worked on in northern British Columbia back when I was in Canada. And it's basically got ocean floor that's been pushed up into the Rocky Mountains. It's got coniferous forests, which generally produce acid soils, but in this case, it's actually pH 8 because the ultramafic rock, which is magnesium rich, is reacting and is weathering, putting magnesium into solution. We looked at microbial mats, so photosynthetic biofilms, similar to stromatolites. And what we found was we produced hydromagnesite as well as magnesite with the bacteria. Abiotically, we could produce a little bit of nesquihonite, which is a magnesium carbonate that's hydrated, but it's just an evaporite. And so if it rains, it dissolves again. The hydromagnesite that formed wouldn't dissolve. That the equilibrium is against that. And so there's a natural hydromagnesite deposit in northern British Columbia in Atlan, and it's biogenic. Yeah, so magnesium works just as well and is easier to put into solution than calcium. Calcium precipitates much more readily than magnesium. Yeah, but it will carbonate, yes. Yeah, so the challenge is certainly in Australia is quarantine. They are strict. And so our South Africa work is happening in South Africa, and our Brazil work is happening in Brazil. We bring small quantities into the lab to run kind of benchtop models on systems, things that we can control and autoclave and kill when we need to. But for scale up, right? That's kind of beyond. Alejandro may argue against that, but when you want to go to 10 meters or 100 meters, that's not something to do in a university lab, certainly not in a microbiology lab. That's something that we need to partner with industry, right? They're used to moving entire mountains, right, to mine out material. And so if we want to get to that scale, we've got to partner with industry and try it under real field conditions, which aren't always kind.